In this third part of topic 2, we are going to be briefly introduced to cell physiology, primarily the transport across the cell membrane. Now, before we start, we need to know some basic terms such as what is a solution. A solution is when two or more components are mixed homogeneously. For example, if you mix salt with water, it mixes uniformly or homogeneously. So it's a solution of salt and water. In the solution, there is a solvent and a solute, which are the two components. So when you are mixing salt with water, the solvent would be the water or the liquid part, while the solute is the salt, which is present in smaller quantity. Now, in a cell, what are the solutions or what are the liquid parts? There are two main liquid parts, the ICF and the ECF. The ICF is all of the fluid inside the cell. So it is present in the nucleoplasm and the cytosol. While the fluid that is present outside of the cell, that means in the space between two cells, that is known as the extracellular fluid or ECF or interstitial fluid. Since cells are dynamic entities, there is a lot of membrane transport going on. That means substances are moving into and out of the cells, and all of this is happening across the cell membrane. And cell membranes have a very important property. They are selectively permeable. That means they allow certain things to pass and also stop certain other things from moving across the membrane. So they are being choosy or selective. There are two main types of transport that is occurring across the membrane. The two types are passive transport and active transport. Passive transport means when there is no energy involved. Okay, that means no ATP is being broken down to release energy. While active transport is where the process requires energy. That means ATP will be broken down to release energy. Both passive and active transports are further subdivided into different types of transport. Passive transport is divided into four types: diffusion, osmosis, facilitated diffusion, and filtration, while active transport is of two types: solute pumping and vesicular transport. Vesicular transport means transport involving a vesicle. And vesicular transport can be endocytosis or exocytosis. We will start with passive transport, that means no energy is required, and the first type of passive transport is diffusion. Diffusion is defined as the movement of molecules, that means solute molecules, moving from an area of high concentration to an area of low concentration. That means the two areas from where it is moving to where it is moving, they have a difference of concentration, and this is known as a concentration gradient. So things will always move from an area which has a high concentration of that thing to an area which has a low concentration. An example would be if, as your instructor, I stand in front of the class and spray a bottle of perfume in the air. What will happen? Initially, the first row of students can smell the perfume, and then slowly the fragrance of the perfume spreads. So the second row of students will be able to smell it, then the third, fourth, and so on. In this figure, the cell membrane is denoted by the orange line. The nucleus is the blue circular structure. So everything inside of the orange line is the cytoplasm, and the fluid inside is the ICF, while everything that's outside of the orange line is the ECF. Now those gray circular dots that you see are molecules of carbon dioxide. So there is some carbon dioxide in the ICF and some in the ECF. So what do you think? Where is the concentration of carbon dioxide higher? Is it inside the cell or outside? It is inside the cell, right? So since there is higher concentration of carbon dioxide inside the cell, carbon dioxide will slowly start diffusing out of the cell. So you can see the direction of the arrow that carbon dioxide is diffusing from ICF to ECF. The second type of passive transport is osmosis. Osmosis is very similar to diffusion. However, there are two basic differences. In diffusion, it's the movement of solute molecules. In osmosis, it's the movement of the solvent molecules or water molecules. But you see that the molecules are moving from an area of high concentration to an area of low concentration. In this case, it is the concentration of the solvent molecules or the water molecules. And another important part of the definition is that the movement has to occur across a semi or selectively permeable membrane. That means your definition is not complete if you forget that it is the movement of the water molecules across a selectively permeable membrane. In this figure, again, the cell membrane is denoted by the orange line. The nucleus is the big blue round structure inside the orange line. Now there are two types of molecules in this figure. The green one is the solute and the little blue dots are the solvent molecules. Now you see there are more green dots in the ECF and more blue dots in the ICF. That means the inside of the cell has more water or solvent molecules. So for osmosis, it is the movement of the water molecules from a region of higher concentration to a region of lower concentration. That means the water molecules are going to move from inside the cell to outside across this orange semi-permeable or selectively permeable membrane. So this is an example of osmosis. The third type of passive transport is a facilitated diffusion. It is a type of diffusion which is helped or facilitated by a carrier protein. Which kind of carrier protein? Remember the integral proteins inside the plasma membrane? These proteins help to transport a particle across the cell membrane. In facilitated diffusion, if the solutes are too big, okay, or they are insoluble in lipids, then it becomes difficult for them to move across the cell membrane. Here the integral proteins, they help in that they serve as carrier proteins. For example, you can see the glucose molecule being transported by a carrier protein from outside to inside. The fourth and last type of passive transport is called filtration. This is the only type of passive transport which is not regulated by a concentration gradient. Instead, it is controlled by a hydrostatic pressure gradient. That means water and solutes are forced through a membrane because of differences in pressure. The solute moves from a high pressure area to a low pressure area. An example of this type of transport, it occurs in the kidneys when the blood gets filtered and urine is formed. We just learned about the four types of passive transport, and now we are going to learn the active transport. Active transport means where energy is required. That means ATP will be broken down to release energy. Why would certain substances need energy? These substances may be too large to get transported across the cell membrane, or these substances may have to be moved against a concentration gradient. That means from a region of lower concentration to a region of higher concentration. There are two types of active transport. The first one is solute pumping and the second one is vesicular transport. Solute pumping, which is also sometimes referred to as just the active transport, in this kind of active transport, the solutes would be transported using energy by a carrier protein, and it could be opposite to the direction of the concentration gradient. That means from region of low concentration to region of high concentration. A good example is the sodium-potassium exchange pump. Here, for one molecule of ATP that is broken down, 3 sodium ions are kicked out of the cell and 2 potassium ions are brought inside. So sodium is denoted by Na and potassium by the K. The second type of active transport is called a vesicular transport. That means transport using vesicles. Where did you hear of vesicles before? Remember secretory vesicles being made by the Golgi apparatus, where protein was being processed and packaged and transported to wherever it had to go. So when these vesicles are used to transport solutes or nutrients or proteins, that is vesicular transport. Vesicular transport is primarily two types. When something is getting imported into cell, it is known as endocytosis. So endo, remember entry. Endocytosis can again be divided into two types, pinocytosis and phagocytosis. Pinocytosis is equivalent of cell drinking. That means when the vesicles bring liquid or really small molecules into a cell, that is pinocytosis. Phagocytosis is cell eating, when vesicles bring large solid particles into the cell, that is phagocytosis. This figure shows how pinocytosis, which is cell drinking, or phagocytosis, which is cell eating, occurs. Both these types of transports use vesicles. A vesicle can be defined as a membrane-bound sac, and these are bringing things inside the cell in endocytosis. In contrast to endocytosis, exocytosis means export out of cell. Remember exocytosis as exit. So here again vesicles will be used, and these vesicles will transport materials from ICF to ECF. Remember that vesicles play a very important role in transporting of materials inside the cell and also across the cell membrane. There are two types of vesicles that are formed inside the cell. The first one is transport vesicles, which are pinched off from the endoplasmic reticulum, and they take materials from the endoplasmic reticulum to the Golgi apparatus. The second type of vesicles are known as secretory vesicles, and these are formed from the Golgi apparatus, and these type of vesicles would usually take materials towards the cell membrane. This figure shows how the transport vesicles and the secretory vesicles are formed. This is a continuation of the previous slide, and it illustrates the synthesis and packaging of secretions.